Hello and welcome to our second installation of Transcription and Translation. Today we are focusing in on the additional higher level information, which means that everything is going to be a little bit new, new vocab, a little bit more difficult to understand, but hopefully after this lecture you're feeling way more confident in your abilities. So without further ado, let's get started with some brand new terminology. We're talking about the sense and antisense strands of DNA. Now, when we use these terms, you've probably heard them in other capacities, just not with this specific name attached to them. So when we're typically talking about DNA replication, we have a template strand and we have the complementary strand. The template strand is the strand of DNA that has the genes that we are looking for coded into them. The complementary is just the complementary base pairs to that template strand, but it doesn't actually code for anything because it's the opposite letters than what you would need to actually code for, whatever gene it is that we're looking for. So the sense strand is the strand that has the coding, it is the coding strand of DNA, right? It has the genes. In this case, it is this bottom one, okay? This bottom one has the genes. Whereas the antisense, and anti means sort of opposite, like if we're talking about like British lingo, not really lingo, but like the way they talk, they say anti-clockwise instead of counterclockwise because it's the opposite of clockwise. So anti in this situation is like opposite of the sense strand. And that's exactly what it means. It's the opposite of the sense strand. So it is the complementary DNA. It doesn't actually code for the genes. However, when the mRNA uses it as its template strand, it gets the complementary base pairs of this antisense strand, which is the same as the sense strand, which means that the antisense strand is used as the template for going through transcription because the nucleotide base order that is created from the antisense strand is the same as the sense strand of DNA, which means that the strand of mRNA that is being created is the strand that has the correct coding for the genes. I know, that's a lot, and I hope that that was clear in my explanation, but we're going to talk way more about this in class, but the sense and antisense is going to be like kind of important to understand as we go through this. So let's talk about protein synthesis just as like a very general overview. So transcription, right? RNA polymerase makes mRNA molecule that's complementary to the antisense strand of DNA. Because when it makes itself complementary to the antisense, it makes itself identical, except U's are there instead of T's, to the sense strand of DNA, which is what we want because that's where the genes are. In translation, we have mRNA being sent out of the nucleus, attaching to ribosomes, who then read the codons and correspond with anticodons to build a polypeptide or to build an amino acid chain. Both of these processes are carried out in a 5' to 3' direction. So transcription, we have a few specific areas, right? We have the promoter area, which is where the RNA polymerase is attaching. Then we have the coding region, which is the transcribe section, and then the termination sequence, which is obviously the end of what is being transcribed. So transcription is the gene results, transcript of the gene results in an mRNA molecule, which is then sent out of the nucleus and into the ribosome so that it can be translated. RNA polymerase moves along the antisense strand of DNA. I know that I'm really focusing in on this, but like I really need you to understand the difference between antisense and sense strand. So RNA polymerase moves along the antisense strand, attaching at the promoter region and unwinding that DNA. Once it unwinds that DNA, it is now free to use free nucleoside triphosphates to make that strand of mRNA based on that unlocked, sort of untwisted area of DNA. mRNA is then elongated until the ribosome reaches the terminator region, right? And so then all these free nucleoside triphosphates are found in the nucleus, taken in by RNA polymerase, and 2 phosphates are removed from that. When they are removed, they are then added onto the 3' end of the growing mRNA strand. And you can see that you are just continuously removing phosphates, adding these nucleotides together, and you are building this mRNA strand, which will then leave the nucleus and go out into the cell itself. Now, exons and introns are 2 additional terms that I think you've heard before, but maybe like you just don't really know what they are. But they're different areas of DNA. So introns are non-coding regions of DNA that need to be removed before translation. And then exons are where the actual genes are, okay? So when you have this area where you have like a coding area, and then a non-coding area, then a coding area, and then a non-coding area, and then exon 3 would be a coding area. And so what happens is you have these splicing enzymes that come in and cut where that intron is, and they sort of bend this whole section together and eventually just like splice out the intron that was there because it's a non-coding region, and then you're left with just the exons themselves, which is now what we actually want. That's all we care about. Once we're through in the nucleus, we move out into the cell itself and we move into ribosomes. And the type of RNA that we're now focusing on is tRNA. And the function of tRNA is just to bring amino acids to this ribosome so that we can continue to build and grow a polypeptide in the correct location. So what this looks like is you have a clover structure, and you see like the red, you see how they have these like little blue dots? Those are hydrogen bonds bonding complementary base pairs to each other. When you get to the outside like loops, those are not base paired. They're not connected, they're just sort of loops. And then you have at the bottom, which is where like this blue part is, on the bottom you have the anticodon, which this part is where the tRNA is going to connect to the mRNA. And at the top, like above this red part, is where you have the amino acid attaching to the tRNA. So tRNA activation, as we've said a few times, tRNA delivers amino acids to the growing polypeptide chain. It picks up new amino acids when activated by a specific tRNA activating enzyme. You don't need to know anything more than that, except that it uses ATP so that you can actually connect the amino acid to the tRNA. There are 20 of these enzymes because there are 20 amino acids. So there is a specific enzyme that corresponds to a specific amino acid. That way, the tRNA has the exact amino acid that it codes for the anticodon and up here to connect to the amino acid. The ribosome, our favorite structure, maybe. It's my favorite, though. So ribosomes is where, like, the magic happens here. It's made of protein and ribosomal RNA, or rRNA. When we're talking about this, the formation of polypeptides, the process of going through transcription and translation, it doesn't happen with just one ribosome. That would take literal ages. That would take forever. What happens is multiple ribosomes work on the same mRNA at the same time to just be more efficient. So this is called polysomes, right? Many ribosomes in this case. Now you should know this, but if you don't, that's okay. There are 2 types of ribosomes. There are bound ribosomes and free ribosomes. Bound ribosomes are those found on the rough endoplasmic reticulum, and whatever they are producing is produced for export out of the cell. Okay? Proteins produced by the free ribosomes, which are the ribosomes that are just floating around in the cytoplasm, the proteins produced for that are used within the cell. So bound ribosomes is for out of the cell, free ribosomes are for within the cell. There are 4 main parts to translation. There's initiation, elongation, translocation, and termination. And I think just from those terms alone, you probably know what these mean. Initiation is when the first tRNA molecules are brought into the mRNA and they bind together. It is always with methionine, or AUG, with that codon. Elongation is the process of forming that peptide bond between amino acids, making a polypeptide, right? You have tRNA binding, the peptide bond formation, the amino acids are being added. Translocation is the ribosome releasing used tRNA, moves along the mRNA to the next codon, and then the next amino acid is brought in, and it continues and continues and continues, but the ribosome is moving in this one. And then termination is when a stop codon is reached, and so everything lets go of that mRNA, that polypeptide is complete, and we move on to a new strand of mRNA. This is just an image showing you this. So initiation, you have an amino acid being brought in. Elongation, you have the formation of these amino acids continuing to be built into a polypeptide. Translocation is the mRNA is being moved, the amino acids are moving down the mRNA. And then termination is when you reach that stop codon and we're done with creating this polypeptide. Just to further list out exactly what happens in initiation, mRNA binds to the ribosomal subunit at the binding site. Methionine binds to the start codon. It is never anything else. It is always methionine. Then the large ribosomal subunit attaches, and the tRNA with the anticodon corresponding to the next codon attaches to that. Okay, so we start with methionine, and then it could be anything. It could be any of the remaining amino acids, but that will connect next. And then you form that peptide bond between the amino acids, and elongation continues in a 5' to 3' direction. So you start with methionine, you move to the next amino acid. When the next anticodon corresponds to the mRNA, with its complementary base pairs, and it will continue to move through until it reaches a stop codon, right? So we have our start codon here, methionine, it's always methionine, and then we continue on, and we will bring in a new anticodon, it will connect, and then these will bind together, creating a polypeptide, and we move on to elongation. Eventually, we're going to hit that stop codon, and it's called termination. So once it reaches the stop codon, it will release whatever factor was attached to it. tRNA is released to find another amino acid because remember, at the top it has that binding site, and it will use an enzyme to bind to another amino acid once it finds it. So it'll find another amino acid, and the polypeptide is released. We have a free polypeptide, right? This protein can be used. And the components of the ribosome break apart because all of this can be reused over and over again as we continue to translate whatever genes we're trying to translate. Now, the last bit that we're going to talk about is gene expression. We've talked this whole time so far about how genes are produced, how we go from DNA to RNA, RNA to protein, but then what? Right? That's what this part's about. So gene expression is regulated by two groups of protein that bind to specific base sequences in DNA. Okay, they're transcription factors and regulatory proteins. The transcription factors are at the promoter site. The RNA polymerase can't initiate transcription without these factors. So if they're not there, we're not expressing any genes. And then the regulatory proteins are outside of the promoter, and they can be enhancers or silencers. I bet you can figure out what that means, right? Activator proteins bind to enhancer sites to increase the rate of transcription, and repressor proteins bind to silencer sequences and decrease the rate of transcription, right? Enhancer enhances, silencer silences. Now, there's also the environment. And the environment can influence gene expression as well. Chemical signals within the cell can trigger changes in levels of regulatory proteins or transcription factors. Okay? So, although it's not specifically in the gene, you can get signals from chemicals within the body depending on where you are in your environment, right? So some examples are humans can produce different amounts of melanin depending on light exposure. Certain species of fish and reptile can change gender in response to their availability of mates. If there's a low availability of females, some reptiles can change gender so that they can continue to reproduce. And then Himalayan rabbits produce different fur pigments depending on the temperature, which is pretty interesting. So the last thing we're going to talk about is epigenetics, which still deals with gene expression, but it's just a fancy term for it. And this is a study of changes in phenotype as a result of variations in gene expression levels. So this is influenced by heritability, but is not genetically predetermined, right? So you can be influenced by your parents, you can be influenced by your grandparents, but it's not predetermined. So identical twins might still have different methylation patterns. And environmental factors may influence the level of DNA methylation that happens within the body. There are two ways to do this, okay? And this is based off of nucleosomes. So nucleosomes help regulate transcription, right, in two ways, acetylation and methylation. So if you add an acetyl group, which is what acetylation is, to the tail of a nucleosome, it makes DNA less tightly coiled, right? It looks like this, right? It's a little bit loose, which increases your transcription. Because it's less tightly coiled, you can reach those genes more easily. But doing the opposite, adding a methyl group or going through methylation, makes the DNA more coiled and it reduces the transcription because the genes are so tightly coiled you can't get to them. So acetylation, more transcription. Methylation, less transcription. Direct methylation. Direct methylation is when you're increasing the methylation of DNA, which causes a decrease in gene expression, which makes sense, right? Because as you increase methylation, you're more tightly twisting around that nucleosome, which means that those genes are way harder to even get to. So you're going to decrease the amount of gene expression that can happen just because you can't get to the genes, right? You can't get those transcription factors in there. So genes that are not transcribed like regularly tend to exhibit more DNA methylation, right? Genes that are actively transcribed and continuously transcribed throughout life, they're going to be easier to get to, easier to untwist and unwind so that you can actually transcribe it. But when you are doing direct methylation, it's typically for genes that you're not transcribing ever, very rarely. So there are some methylation factors that impact whether the DNA is going to be methylated or not. So like if a woman is pregnant, the diet that they are eating throughout their pregnancy will determine what genes are more likely to be transcribed or not, depending on what they're putting into their body. When you're an infant, what you're exposed to as an infant is going to impact. If you're exposed to a lot of microbes, then maybe you need a higher amount of genes that are going to impact your immune system. Whereas if you're like kept in a bubble, you're not exposed to anything, so your body doesn't know that it needs to make those kinds of things. Stuff like that. So direct methylation and acetylation will just typically determine what genes are continuously being expressed or kept tightly wound and not being expressed. We're going to practice this more in class, but great job, and I hope that if you had any questions, you go back and you re-listen to this lecture.